we know we've moved quite a bit down here. So. Okay. All right, so if you're following along, we're pretty much we're on page 69, textbook. The theories of explaining antibody diversity. So there's been several, and if you read, it's a real good history in your text, gives you a little background as to where we were and where we come to. So you'll see stuff like the side chain theory, and it was by Ehrlich in the early 1900s. And he said, well, how about let's postulate certain cells have specific surface receptors for the antigen that were present before contact with the antigen occurred. So he kind of came up with this idea, which proves right, that the cell actually has receptors to the antigens, and the antigens can bind to those, right? We know those as, right? Hopefully that makes sense. If the antigen is introduced, it combines with a proper receptor, breaks off, and enters the circulation as an antibody molecule. That's not quite how it works, right? It doesn't break off like with the antibody and then moves on. So he postulated this, and it could be repeated in further contact with the antigen. So that's the side chain theory. And then in the 50s, we had Journey and Burnett's clonal selection of antibody formation, where they said the lymphocytes are genetically pre-programmed to produce one type of immunoglobulin, which right? So this was kind of the breakthrough of saying, you know, there's something that our cells know. You know, the cells, once they get exposed to an antigen, they know how to produce to that antigen. So there was this one-on-one concept, where a specific antigen finds a particular cell, that cell can respond, causing proliferation, but this would require a large number of genes, and they weren't sure if that's what was happening or what could happen, but it kind of led to the next step, which was in 65 by Dreyer and Bennett, when they said there were constant and variable portions of the immunoglobulin chains coded for separate genes. So that's our concept of that, you know, immunoglobulin, the constant part is the same for everybody, but the variable part is the one that's going to be specific for the antigens. Make sense? So you look at these years, right? You know, I was just born 2 years after that, so you know my age. But you guys don't even know what the, do y'all remember the 1900s? Do y'all remember that time period?
Yeah.
Okay, y'all were around in 1999?
Yep.
No?
Yes.
You know, there was a song, you know. So what is going on here? And if you're having trouble, this is where I go back and refer to handwritten tutorials. I've watched these, and I used to run them through class. We had enough time where we'd actually open up this video, run it for 10 minutes, and watch it in class. But I highly recommend that you watch his gene coding for immunoglobulins and how that works, because we're going to see just a sniblet or a snapshot in the text on this. Okay, so you got to be ready for that. And the snapshot you're going to see is on 71. So he'll go through that and show you what's going on and how it's rearranged. But we have this, chromosomes contain building blocks. We understand that. The human immunoglobulin gene are found in 3 unlinked clusters. Okay, there's the H chain in chromosome 14. There's the kappa chain in chromosome 2, and there's the lambda chain in chromosome 22. So you may see a question, you know, that puts in you know, always there's a hand that goes up about this time and says, do we need to know the chromosome number? And I always say, well, if it's up here and we have a number, we kind of need to start putting that in a memory bank so we can understand where these are located. You might get that. You might get the human immunoglobulin gene. It's what is contained in 3 chromosomes. Which ones? Okay, simple question. puts the number out there. Not easy to remember 14, 2, and 22. I don't know how you can go about remembering that. My favorite number when I played sports was 22, okay? I don't know where 14 and 2 come, maybe you can do that. Maybe you can associate your softball years, your glory years in softball with your jersey number. But that is association with human immunoglobulin. H chain, lambda chain, kappa chain.
What page do you start on?
We're about 70. We're in there. You'll see chromosome 14 up there at the second column. You'll see this in 70 in the first column at the bottom. So if there's more than one gene controls the synthesis, then we've got to have something different, right? We've got to have, and if you look at the coding, if you look at page 71, you will see coding for immunoglobulin H chains. There's 4 separate regions on chromosome 14. There's, for the H chain, there is the D and J region, okay? There is VD and J combination. So you see all of that starts off really long, and then there's some changes that occur. So we got this DNA chromosome, and you'll see a variable out there, you'll see a D, you'll see a J, and you'll see a constant. So we really call the variable region VDJ, okay? Which means that's where the rearrangement occurs. And you see there's some excisions, there's joining of D1, D2, and J3. V, part of V comes out, and we end up with, if you want to say, we come down here to the very end, and we get a V2, D2, J3, and a constant mu, which means for IgM, okay? So, again, I don't want to get into the genetics. I don't want to spend weeks on trying to figure out if you understand what's going on. But you do need to know that there's the H chain, right, of the immunoglobulin found on chromosome, which one? H is located where? 14. There's a variable region. The variable has variable D and J, VDJ. The constant region is C, okay? The light chain lacks the D region, okay, so it's not there. So the light chain would only have who? B and J. So I know this is weird to think about. I know you don't have much in genetics. This is not a genetic class. But what we're showing is the actual principle behind how an antibody becomes variable. How an antibody becomes its one and only for the antigen. How that works. Okay, so all these genes can change expression. So, you get exposed to an antigen, the cell does this change. The cell changes by genetics, randomly selection process. These individual segments are joined to commit the lymphocyte to making antibody to a single specificity. Well, that's our main goal, is that antibody set, the lymphocyte that's producing the antibody went through this change, okay, so that it could be variable. So all these different genetic combinations gives us our, how that happens. Now, it explains it. It says that, yes, it's true that there is one lymphocyte that produces antibodies against one specific antigen. Now, those antigens can have similarities, okay? So if I've got bacterial antigens, when we talk about gram-negative bacteria, they have similarities that we have other, this cell was produced against E. coli, but it also may have some effect on another gram-negative rod, right? So keep that in mind too. It doesn't have to switch from, say, an E. coli to a Pseudomonas if they're both gram-negative. So there could be some similarities in their structure that these could zone in on. So here's that heavy chain rearrangement. Remember this, so if you saw this top part, the key here is to know that if we have V, D, and J on the chromosome, that you're looking at what? Heavy chain rearrangement. Remember, the heavy chain has variable region too. Don't forget that. Don't think that just at the top it's variable without a heavy chain variable. Because that heavy chain goes all the way to the top. It's the light chain that doesn't go to the bottom, right? Major Y. I've been practicing drawing the IgG and the IgM. Now, and you went home and showed your kids what an immunoglobulin looks like to impress them about your schooling that you're getting. Like, yes, I know you know 2 + 2, but look what I know. I know immunoglobulins, right? You don't do that?
Not regularly.
Oh, seems like fun, doesn't it? You were wanting me to give you some kind of fun idea for home, and what's more fun than drawing immunoglobulins and coloring, right? Isn't that neat? All right, so you should know that this is VDJ. That's the variable region. This is the constant region. And we're going to rearrange this depending on what was needed until we get all the way down to translation and we have what we want, okay? So that's going on in those lymphocytes. So that would be a heavy chain. This would be the light chain because who's missing? D. D's not here. So in the VDJ, now we've lost D for the light chain rearrangement. We only have D and J. We still have the constant region. They're still rearranging, okay? And we come down here to the light chain protein, which is BJC. And I used to get real wrapped up here and go, do y'all not know what translation and transcription? And we just kind of went ahead and just said, no, you don't, right? And that's up to you if you want to understand this better, right? Because we know that you've had that since high school and you forgot it and you just never wanted to learn the process of how genes work. But you hopefully do know DNA and RNA. But you're going to have to know that for the rest of the year. Now, the transcription and translation, hopefully you can put those in a line of how it goes. And we'll be good. And you hopefully know transcription of a DNA gives you messenger RNA, which is then translated, okay, into the protein by the ribosomes. All that making sense? Some of you are going, yeah, I got that. I knew that since 8th grade. Some of you are like, I knew I should have listened to DNA RNA talks. All right, so that's how it works. Again, handwritten tutorials. If you just like, I just don't get it, he'll draw it for you. He'll show you how those splice in and out. Okay, so he gives you a little thing instead of just here, here, and here. He'll actually train it. He'll actually, draw them different and talk about how those are leaving, and when that leaves, then that changes what we end up with. Okay, you only going to come down to a certain number of genes there at the end. Monoclonal antibodies. This is exciting. I said y'all wanted something exciting for today because he put you to sleep. So monoclonal antibodies, this is where we're doing. This is new stuff. This is treatment. This is working stuff, okay? You'll have family members that have done, hopefully, if they're suffering from an autoimmune disease, they've benefited from the monoclonal antibody. We do it for testing in school, and all your little blood bank antibodies you'll get next semester are going to be done with a monoclonal. That's how they got them produced so big and put them in a bottle for you to drop into the tube and go, oh, look, the glutination. Right? You'll be doing that next semester. But for us, therapeutic treatment is key. Okay, so we can treat diseases now because we figured out how to get monoclonal antibodies produced. And we can do passive, hey, we talked about that with COVID. We said, if I've got the antibody, I can be passively given to somebody else with my plasma, and it might help them get off the ventilator. Maybe, right? But we now can produce targeted antibodies, right? And this can be for cancer cells. This can be for out-of-control B cells, which got one of my stories. I can tell you about one of the ones that is in here, one of the treatments. We're going to tell that to finish it up, this chapter. They're derived from a single parent antibody producing cell. So we still have that main concept of here's the antigen. We've got an antibody producing cell that can produce an antibody, but it can't produce enough. Okay, we took it out and we said, hey, produce all you can and we'll use your antibodies to treat. We'd never have enough to do it. So the concept, this is where we use a cancer cell, right? So we do a hybridoma is where we take a malignant cell and we combine the good part of an antibody producer with the good part of a cancer cell, which is I can replicate over and over before you blink, right? Kind of concept, which is really good because that's a machine producing antibody. That's what we want. And that's what happens. So we have this where we form a hybridoma, where we fuse an activated B cell. Now you understand that. We've talked about the B cells enough, and we've talked about the maturation. We've talked about the activating of the B cell and how that happens. And what we're going to do is we're going to fuse it to a laboratory-grown myeloma cell that cannot make its own DNA because it's deficient of HGPRT. I love when we shorten those up because then I get to ask you guys, what is HGPRT? So now you're quickly going through this and you're like, I had a micro test yesterday.
That's a big word.
It's a big word. Well, try it. I love big words when you try to say them.
Xanthine, guanine, phosphor, lyse, transferase.
Yes, great. Great job. So, because it's deficient in that, can't make its own DNA, so guess what we're going to do? We're going to give it that B cell DNA. And then once that myeloma cell gets some new DNA, then it's like, phew, look what I can do. We're moving. Okay? So how do we get that? We immunize, and we have a picture. I don't have the picture. You have the picture in your book. Sorry. You have the picture. We have the word. So you look on your page 73. We have the picture. We're going to take a mouse, and we're going to immunize a mouse. So we call that murine. So anytime you see murine, it's coming from a mouse. So when you get your little antibodies next semester, we're going to have some today. When you get that, look at the bottle and see what's printed on there and see where that monoclonal antibody came from that you're working with out of a bottle. Okay, it's really fun. What we do is we immunize the mouse against the antigen. Okay, we know which antigen. We're going to immunize the mouse. The mouse is going to start producing antibody against it. So those activated B cells are mouse B cells. You got it? So we get those out by harvesting its spleen. We know that because we know the spleen was the source of where B cells ended up. We combine the spleen cells with the myeloma cells in the presence of PEG. You might know what PEG is. Polyethylene glycol. It's one of those words you're going to hear over and over. Okay? And then we're going to select the fused cells and screening for the presence of the desired antibody. Okay, so if you look on your picture, you'll see they split that up. So you got your mouse spleen cells with your myeloma cells cultured. These are cell cultures, and we're going to fuse those together in the presence of PEG. And PEG's what again? Polyethylene glycol. Okay, and we're going to place in HAT restrictive media. And HAT, I saw it a while ago. What is HAT? Hypoxyanthine aminoterine thymidine. That's what we call a HAT. Okay, another chemical there. Hybridoma cells grow in that, and the spleen cells, if they're by themselves, die. And the myeloma, if they didn't fuse, they die. So the only ones that grow are the hybridoma cells in the HAT. Those hybridoma cells now can secrete antibodies to a level that we can gather and bottle and market and ship to you and charge your school money for them or your hospital for them, and we can make a business out of that. Okay, so isn't that exciting? You know, hematology was, you know, what hematology is. But this is what? I could actually take my degree and go where? Go to work, right? I could go work at one of these companies that's making millions of dollars based on monoclonal antibody production. And I now know from one little lecture, one little page in my book, how that works. All right.
I have a question that kind of pertains to it.
Sure.
I'm going to get my blood taken, just my blood,
Mm-hmm.
Sunday. Do they still test for the COVID antibodies
Yes.
whenever I
Yes, they're still testing for
Because
it.
at first they had me for platelets, but I didn't want to do that. I just wanted to do blood because I think that's
Yeah, they'll
pretty
need
much-
your plasma to test for antibody.
So they can.
Yeah, if you're donating a unit,
Yeah.
they'll do it. Yeah. All right. One of the stories I get to tell, and we'll finish up chapter 5 with this story as I'm switching gears here. And we'll stop sharing that one, and we'll pick up number 6. Where did number 6 go? I don't think I've opened number 6 yet, have I? There's number 6. Okay. Now I can go share. Zoom, y'all doing good? Share number 6. That's 5. Where did 6 go? Did I not get it open? Share screen. Maybe I should close 5. I don't know. Close 5. That brings up 6 minutes. Nope. Maybe I should close that. Try again. There we go. All right. So we're sharing now, and we'll get started on the first one. Okay, so cytokines. But here's the story before we leave chapter 5. Okay, so my wife, she loves that. I always tell her, I'm like, hey, you ran my lecture today. And I always tell her a story because she has autoimmunity for thyroid, which is she has Hashimoto's antibody against her thyroid gland, which causes her to be hypothyroid. So she has to take Synthroid, all that good stuff. But that led to something else. What that led to was she was having issues abdominally. All of a sudden she had these sharp pains underneath and just was going to get tested for whatever. So I went to the ER, which is the worst place to go because they don't know what's going on, you know, and it went from, well, where you're having a stroke to you're having a heart attack to we really don't know what it is, but you're getting better, right? So she ended up going to a rheumatologist. I think it's rheumatologist. Maybe it was an endocrinologist. One of the two led her to the endocrinologist where we did a whole group of tests, right? We did the whole menu of testing. And one of those was, I feel like she's probably leaning toward lupus. Okay, so everything was shooting toward lupus, but we'll wait on the results and we're going to send you to a nephrologist. Is that the kidney doctor? So we went to the kidney doctor, long story, short answer. So did a biopsy of her kidney, okay? Because she was spilling protein in her urine at a high enough level that the kidneys were being targeted. And something was wrong with the kidney. So we had all this roundabout, we ended up at the kidney doctor. And after the biopsy, which it actually got sent to Little Rock for testing, which is pretty neat, I guess. And what they diagnosed her with was something called onco. Okay, so it's acute inflammation, autoimmune, but what's happening is she has this antibody attacking the lining of her vessels. Okay, so that was it. So if you look at the clinical application, you'll see the drug that she took. Okay, so the drug that she took was called Rituximab. And usually these antibody drugs have AB on them. So you see it down on 72, you see Rituximab, Rituximab, and that AB usually means it's an antibody, an immunoglobulin treatment. So what she took 3 of these treatments over a period of 6 months, got an infusion of this antibody that was going after her B cells. She had these B cells that were producing a wrong kind of antibody, didn't want that produced. It was actually an attack. So we designed this Rituximab, which is just a monoclonal antibody, given through an IV that circulates through her body and finds that B cell and attaches. So the body started removing her B cells, but of course this is like a cancer treatment. Thus, it really put her in a state of, you need to be careful not to get something else while your body's B cells are being attacked. Because we don't know if it was attacking the right ones. We hoped it was, but we wasn't for sure if it was attacking the wrong ones. Has she ever had hepatitis? Has she ever had TB? All those good diseases that can come back if you take your antibodies out. Okay, so long story short, we've got this Rituximab on board, and now she is called in remission for onca because we were able to treat for the B cell. Okay, so you'll see things like when we talk about rheumatoid arthritis and lupus, there's treatments building every day that can go after the body's own antibodies. I know that's weird, but that's how we're treating autoimmunity. We're treating cancer that way. Once we do a mark on a cancer cell, we can produce an antibody against it. Infuse it, let it go after those cells, okay? Try to remove those cells as much as possible. So this is kind of the new line of treatment. So it's more of an antibody design treatment. We see Crohn's disease is being treated this way, rheumatoid arthritis. So you see these names that we're going to revisit when we get to them in the autoimmune chapters, and we'll revisit these treatments. But that is one of those monoclonal antibodies that was useful for us, that actually put a pretty severe, I mean, it wasn't like, oh my gosh, but if her kidneys kept getting damaged, then yeah, it would have been, oh my gosh, right? So luckily we were able to find that early and limit the damage to the kidneys. All right, so that's my little story involving my wife and bringing her into the lecture today. So cytokines, yay, cytokines. We finally made it to chapter 6. Whew. Okay, so it gets a little easier for me because basically what we're just talking about is re-talking over some of the things that we've already talked about and then assigning a player. So we're going to assign new players that you've heard of. We talked about them on day 1. Now we're putting them into the context of these are the actual cytokines, and we're going to put them in who they are and what they do and how they help us, right? So these are helpful, and then there's some that are too helpful, and we have to have ones that counter that help. So cytokines, these are the communicators. Okay, we have them in the innate immunity and we have them in the adaptive immunity. So they play a role in both. And we've had them said in both, and we've had them on charts in both. So they're small soluble proteins. They can regulate the innate immunity, the adaptive immunity. They're induced in response to stimuli. So the bacteria bring about the cytokines. Flagellum, the parts of the bacteria for movement, bring about the cytokines. Other bacterial products, their toxins that they produce, bring about the cytokines. Okay? So they work this way, and this is always fun because this is, hey, we didn't know we really have to talk about synergy, pletropy, redundancy, and antagonism. So we have some definitions that we have to know for this one. So synergy just means that you can have all the cytokines working together. They act together. They build up. Hey, if I'm here, you're here. Now we can increase the body's response to an antigen. Pleiotropy is one cytokine has many actions. So I can say, I do this, but I also can do this. And watch me now, I can even do this. So they have many roles they can play. So they're not just designed for one thing. Redundancy. Different cytokines have the same function. This is kind of like, I can do that, and so can I. And he can too, and I've got another one over here. This cell produces this one, and that can do this. So we see all this kind of role play. And so it's kind of like a play when you think about it. And then we have our antagonists where they counteract each other. So you don't need to be doing that. It's time to stop, so I'll come in and we'll calm everything down. So this is kind of like the opera, it's the play, it's the orchestra, whatever you want to say when it comes to cytokines, because that's what happens when we talk about what they do. So this is from your book. This is on page 79, and this gives you an idea. You know, this is the target cell response. It sees by itself, but then you can have A come in and antagonize it and bring down that response. You can have B alone, but when you get with A, its effect goes up, so the response goes up when they're together. So you see that B alone is here and A alone is there, but when they're there at the same time, look where we can go. So that's just to try to put some, a picture to what we've been saying with the roles that they play. So here is the role of a cytokine. They can get leukocytes and other cells to grow or to not grow. So they influence regulation of growth, they influence differentiation, and they influence gene expression. And we're going to see that play out in the next few slides. So here is an autocrine stimulation, and that's a new word. I don't think we've said autocrine in the lecture. What is autocrine? Yes. So I secrete something for myself. I'm selfish. I'm not going to let what I need, I'm going to produce what I need. I'm all alone. I don't need anybody else's help. So an autocrine would be what I produce, I use. Okay, so it makes me better. Paracrine.
Affecting a target cell in close proximity.
Paracrine, affecting a target cell in close proximity. So I'm pretty close to where I'm affecting. So my influence is there for something else close by. And then occasionally it says we have a systemic or endocrine activity, which means is what? I'm in the system, I'm having to go far away to make an influence. But occasionally we can do that. And you see the role here. If you want to see who's playing here, I see the brain. Okay, the brain has a role because it has the hypothalamus. And if you haven't read ahead already, the hypothalamus is our thermostat. And you know how fever is being used every day when you go to lab. We check your forehead for fever. Okay, so we get, here's our antigen presenting cell that we're familiar with. This is our T helper 2 cell. This is a neutrophil out here. Okay, and you can see paracrine being released from an antigen presenting cell, affecting these other macrophage. That looks like a dendritic cell. You see an autocrine where what I'm producing is an antigen presenting cell. I'm using it, not letting it go anywhere else. Endocrine is it goes away from me, goes to other places like the brain and the liver, far away. Okay, so that's our 3 words put into a picture for you. So cytokines, multiple actions. And if you read ahead, you've heard kind of the history of where cytokines got their names. Okay, and that's always fun to talk about. That's basically they get their name on where their influence is. So If I affect white blood cells, I'm a leukocyne. If I affect macrophages, I'm a macrokine. We call them cytokines. So have multiple actions, widespread distribution, because there's receptors on many cells for the cytokines. So we have receptors all over our body for cytokines. Ability to alter expression of numerous genes, may share properties, have overlapping effects, and can determine whether the host will be able to mount an effective defense. Now, one of the things that you've heard, hopefully, about COVID is what? What is the big concern with COVID? And we have that on our next page when we see a clinical correlation. Their correlation in your text is with Ebola. We don't need to know about Ebola right now because we have COVID right now. What is the big concern with COVID in the immune system? What causes the patient to pass? If they got underlying issues that really ramp up when they get COVID, so what would cause your kidneys to go ahead and fail, even though you're okay with kidney failure? You're on dialysis, you're good, right? All of a sudden you get COVID on top of it.
Are you talking about a profuse release of inflammatory cytokines?
Yeah, but we have another name for that. You've got the right definition, but it's known as something like called cytokine storm. Okay? That's the biggest concern with patients with COVID, is that all of a sudden they get all this, right? Get all this widespread recepting going on, and all of a sudden the body just dumps cytokine. Produces way too much. So it puts the patient in a dire straits, puts the patient down where their lungs are not functioning as well. Their kidneys are definitely not functioning as well. Because of the immune response to COVID, this cytokine storm was supposed to be the big thing everybody worried about. Okay? And one that has been designated, which we don't have it in the text today, we have the name, is Interleukin 6. So you see that on page 81. Interleukin 6 is the key cytokine with COVID. And some labs are testing for interleukin 6, and it basically means if we see it rising, the patient's going to be in trouble pretty soon. Okay, so we keep a monitor on IL-6, and we see that we want to see it coming down if it went up. We want to see it stay low. So we do like a 6 to 12 hour check of it every day on our hospitalized patient. The lab that told me this, the lab that informed me of IL-6, was a lab that thought ahead and ordered the platform to test for IL-6. So you'll come to a lab probably inside the city that probably can't do IL-6. They can't test it. They'd have to send it off. Well, anytime you have to send something away from the lab, the hospital where you are, that delays the response. Okay? So it'd be great if I could run IL-6s in my lab, because then I could monitor it. It's hard to monitor IL-6 if I got to send it to Memphis every day, right? And I may not get my results until 8 hours after they pick it up. And they pick it up at 3 in the afternoon. I might have drawn it at 8 that morning, so you can see that I might not get my results until 8 the next day. So that's the kind of thing that we have to fight against, because so many hospital labs are doing that because it's easier, it's more convenient for them just to have the guy come by and pick it up in the car and take it somewhere else to be tested. So I'll get off my soapbox a little bit and come back, and we'll talk about the major cytokine families. We've heard of these, I think. All right, I think we've talked about most of them, some way, some form. Tumor necrosis factor. All right, you look at my resume, I have tumor necrosis factor on my resume. This was my college project when I was in grad school. Okay, I took tumor necrosis factor, injected into my chickens up in Fayetteville, right? And I waited on an immune response. I've been doing immunology since grad school, right? Doesn't seem, that was, no, that's 1989-ish, 1990. Yeah, anybody remember the 90s, 1990?
Was born in 94.
94,
96.
96. Anybody? Nobody? Wow, you almost out on the 90s. That was back when, I hate to say it this way, but that was back when Arkansas had a good football team. 1990s, 80s. I don't know what it'd be like to go to the school now. Didn't have, like, what do you do? Like, what do you do on Saturday? You go to the game or not go to the game? Anyway, back to tumor necrosis factor. I gave the chickens tumor necrosis factor. The only problem with these, you have bad research, I kind of did. I gave them human necrosis, tumor necrosis factor. So it really didn't change anything with the chicken. Didn't really get the temperature up, didn't get their white cells up. Now I used to do smear. Have you ever done chicken smear, blood smear? They have a lot more white cells than we do. It's a lot easier to count it. You can do a diff like that on the chicken. Like, I got it done, right? Anyway, tumor necrosis factor we're going to talk about coming Monday or Friday. We got class Friday. Interferons. We have some names, and you heard me say gamma because I just associate gamma with interferon. And those goes against the viruses. Okay, so we have virus cytokines. Chemokines, there's a whole bunch of those. They just keep the name chemokines. Transforming growth factor, so you'll see TGF in your text, and you need to know that's not tumor growth factors, that's transforming growth factors. And then CSF is not cerebral spinal fluid, it's actually colony stimulating factor when we're in immunology. Okay. So those are a group of our major cytokine families that we're going to work through. But where we're going to see them is in the innate immune response. You guys are very familiar with the innate immune response because this is what we're born with. All right, we're born with this because we have cytokines that trigger the recruitment of the effector cells. What are the effector cells in the innate immunity? With time for the class. What are the effector cells of the innate immunity?
Are you looking straight at me?
I'm just looking at eyes that look at me. It's usually when all the eyes go down for the lecture material, like they just, oh yeah. They might drink a Coke, just trying to avoid the teacher at this time. What did we learn about the innate? What are we born with? What cells are we born with? You're doing diffs, right? What cells are we born with? Neutrophils, macrophages, right? Those are our innate. Those are the ones that just phagocytize to phagocytize, right? They're not being adaptive. They're just saying, I'm here. But we have influencers over those. Here are all the influencers. Interleukin 1, tumor necrosis factor alpha, interleukin 6, chemokines transforming growth factor beta, interferon alpha and beta. Okay? So that's what it's going to set up. We're going to end on time. And we're going to set up, there's no need to get into these. So we're on page 80. Okay. We have a little story about the Ebola. And then on Friday, we will definitely get through this and get into complement. All right. Hopefully that wasn't boring. Hopefully you got through it. Hopefully you're getting something from the lecture. Zoom land, we'll see you in lab.